So good morning, everyone, and welcome to the webinar. So my name is Marylène Vandevenne, and I'm the manager of the Robotein facility, which is located in Belgium, and which is one of the Instruct-ERIC facility. So very quickly, Robotein is a high-throughput facility specialized in protein characterization using different biophysical approaches. So we perform high-throughput stability measurement assay, high-throughput enzymatic studies, and high-throughput protein interaction measurements. And so Robotein entered the Instruct-ERIC network in 2020, and I'll just give you a quick overview of this network. So Instruct brings together different platforms and facilities located in various institutions all across the European Union. And the main goal is to promote exchange of expertise between the different universities and research centers in the field of structural biology. So any researcher located in the EU university can apply for fundings via the Instruct platform in order to access and be trained at the Instruct-ERIC Center. But you can also work in a remote manner where you can send your sample for analysis to one of the Instruct Center. So I just would like to emphasize a specific project within the Instruct network, which is the ISIDORE consortium, and which promotes research in the field of infectious diseases. Just switch to the next slide. Yep. So Instruct includes services from protein sample preparation, like protein production in various organisms, engineering of antibody fragments, or protein sample preparation for crystallography. So Instruct-ERIC also covers a large range of biomolecular analysis centers, like imaging, biophysical characterization, and mass spectrometry. So finally, Instruct also includes facilities specialized in 3D structure determination by X-ray crystallography, NMR spectroscopy, or cryo-EM. So today we'll have two speakers. The first one is Nikitas Georgiou, who is a PhD student at the National and Kapodistrian University of Athens in Greece. So Nikitas is doing his PhD in the field of pharmaceutical chemistry and uses NMR spectroscopy and in silico studies to identify new chemical compounds that have specific and interesting biochemical effects. So the title of his presentation will be Potential Anti-Inflammatory Drugs. So Nikitas, I'll just stop sharing my screen. There you go, it's all yours.
Okay. I think now that you can see my presentation.
Yes.
Okay. Thank you very much for the invitation. My name is Nikitas Georgiou, and I am a PhD candidate in the Department of Chemistry of the National Kapodistrian University of Athens in Greece. I'm going to present you one part of my PhD thesis, which one part of it was conducted through ISIDORE infrastructure in the University of Helsinki in Finland. The title of my project is Potential Anti-Inflammatory Drugs. My project consists of three parts. The first part was the structure assignment of 4 possible bioactive compounds through NMR spectroscopy, and then the saturation transfer difference NMR spectroscopy with the 5-lipoxygenase enzyme. The second part was the conformation analysis of these compounds through DFT and 2D nose spectroscopy. And the third and last part was the in silico experiments of molecular docking, molecular dynamics experiments in this enzyme. Now I'm going to tell you a few things about the compounds that I studied. The 3 of the 4 compounds belong to the family of halcones. Halcones constitute a large group of bioactive compounds which they are belonging to the flavonoid families. They are abundant in many plants, flowers, fruits, and vegetables. As you can see from the structure, this unsaturated double bond next to the carbonyl group enhance their biological activity because this kind of compounds, they possess a lot of pharmacological actions like antioxidant, anti-cancer, antivirus, or also anti-malarial. Here also you can see an example of a bioactive halcon derivative, xanthohumul, which has been characterized for anti-cancer activity in vitro. The fourth compound that I studied belongs to the family of 4-hydroxy-2-quinolinones. These kind of compounds, they are heterocyclic, and they also process a lot of pharmacological activities like antiviral, antimalarial, and also against SARS-CoV-2 virus. The new about these compounds is that their structure allows them to accept a lot of chemical modifications in order to enhance their biological activity. The enzyme now that I studied belongs to the family of lipoxygenase. Lipoxygenase, they are enzymes that they have an iron in the active center. As you can see here from the picture, the iron is this in purple color. And their main role is to catalyze the oxidation of plants and in arachidonic acid in mammals. Inhibitors of this enzyme now they are very important due to the implication of this enzyme in a lot of pathophysiological disorders and also in many food-related processes. Dysregulation now of this enzyme may lead to a lot of serious diseases related to the inflammation and also in certain type of cancers. Consequently, the development of this kind of inhibitors, they have attracted a lot of attention in the field of medicinal chemistry. And this kind of compound, they're therapeutic targets mainly to the diseases related to the inflammation and also to allergic diseases. Here you can see some known inhibitors of this enzyme lipoxygenase that they can be used as therapeutic targets in a lot of diseases related to the inflammation, like arthritis or asthma. Now we'll proceed to the results of my project. Here you can see the four compounds that I studied. VK1, VK2, and AD26 belong to the family of halcons, and ADVA compound belongs to the family of quinolines that as I told you before. Now, as I mentioned, the first part was the structural assignment of these four compounds. For ADVA compound, the structural assignment was performed in the National Institute of Chemistry in Ljubljana in Slovenia, and for the other three, the halcon derivatives, the structural assignment was performed in the University of Helsinki in Finland. Specifically, homonuclear and heteronuclear NMR experiments were conducted in order to elucidate all these compounds. And specifically, DMSO was used as a solvent. Here you can see the 2D noise spectra that we took for ADMA compound, and here also you can see the strategy that I used in order to elucidate this compound, and I'm going to tell you now in details. As a convenient starting point, proton 281 was used because the integration from the proton spectra was 3, and also because it doesn't correlate with also here. Now, we knew from bibliography and from previous studies that NH protons, they are exchangeable, and for that reason they appear as broad peaks in around 10 ppm, as you can see here. After the identification of proton 10, as you can see from T2 nose, you can see a correlation between a proton which should be proton 10 because it's next to NH, and then proton 10 was confirmed. After proton 10, proton 8, as you can see here also the correlation, proton 8 was identified. After proton 8, proton 7, also from the structure, was assigned. And then from proton 7, proton 3 was assigned. Also, all this that I told you now, you can see here also in arrows. All the remaining protons in the aromatic, on this aromatic ring, they were identified through 2D Gaussian spectroscopy. Also here you can see correlation between proton 21 and proton 19, and for that reason proton 19 was identified. Also all the other protons, they were determined through 2D Gaussian spectra. After the whole identification of the whole protons in this structure, we had to identify the carbons. For the carbons that they had 1J coupling with the protons, they were assigned through 2DHSQC spectra, and for the remaining the carbons that they were carbonyls or quaternary, they were assigned through 2DHNBC spectroscopy. Here you can see the 2DHNBC spectrum, here you can see the 2D Gaussian spectra. as you can see from 2D Georgiou, we can see some correlations between proton 16, proton 17, and proton 17 with proton 18, as I told you before. Now we proceed to the Halcon derivative AD26. Again here, methyl groups, they were used as initial bond because the integration was 6, because we have 6 protons, and because they don't correlate with no other proton in this structure, so they will give single peaks. Also from 2D Nozi, from proton 4 from this methyl, proton 3 was identified, and from proton 5, as you can see here, proton 6 they was identified. Also, all the remaining protons, they were determined through 2D Georgiou and 2D Nozi NMR spectroscopy, and also again the carbons, they were assigned through 2D HSQC and 2D HNBC spectroscopy. Here you can see the overall strategy that I used for the one of the 3 Halcon derivatives, but it was quite similar for the other 2 because, as you can see, the structures are quite similar. And now the next part was to determine some possible conformation of my compounds. When someone does conformation analysis, he can choose 2 different approaches. The first approach is because we knew the correlation. For example, here I have the ADMA compound that we saw from 2D Nozi spectra. We saw a correlation between proton 8 with proton 7 or proton 8 with proton 10. And then we make a conformation and we keep these distances between these protons would keep constrained, and then we optimize this conformation. The second approach is to make different initial conformations of ADMA compound, for example, and then we optimize all of this conformation, and then we take the lowest in energy conformer, and we compare with the correlation from 2D Nozi that we observed. Now in my project, I use the second approach. For the calculations, all the calculations they were conducting through density functional theory, or DFT, and I used BP86 as functional and F2SVP as a basis set in the level of theory. And all these calculations, they were performed through Orca software, but the visualization of the results, it was performed through Georgiou and Avogadro softwares. Here you can see the results. As I told you, I used the second approach where I sketched various initial structure and then I optimized all of this structure. And here you can see the lowest in energy conformance for each compound. For example, here for ADMA compound, we have observed from 2D Nozi correlation between proton 7 and proton 8. And from the conformation, the distance between this proton, it was around 3 Armstrong. So it's very normal to see this correlation to 2D Nozi. Also here in AD26, we observed between proton 5 and proton 6, we observed a correlation to 2D Nozi, and here it was around 4 Armstrong from the conformation. So also again, it's very normal. So the computation and the experimental experiments, they match. Now, the next part was the saturation transfer difference STD NMR spectroscopy. This kind of NMR is a very powerful technique because someone can use it to see some interactions between some possible ligands with one enzyme, and also it has a lot of applications in the drug discovery because someone can see the binding site of a receptor molecule. Also here in the picture, you can see the 850-100 MHz NMR spectrometer, here the white, that we used in the University of Helsinki in Finland. Now, for the experimental part, before I had to make the solution with the whole complex, with the enzyme and the ligand, I had to make the stock solution for each ligand. This was performed, I solved all the ligands in DMSO. After, I had to choose which buffer I have to use for this kind of NMR, and we chose potassium phosphate buffer from previous studies in pH 7. Then we have to determine how much enzyme and how much ligand I have to add in this NMR tube. And now we started from 1 to 50, 1 enzyme and 50 ligand, then 1 to 100, 1 to 300, 1 to 500, but the best was 1 to 1000. That's why, because my ligands, unfortunately, they weren't soluble in D2O, the compounds, and they were soluble only in DMSO. And for that reason, I had to add more and more ligand to see some peaks in the NMR. Now I'm going to explain the results. Here you can see for ADMA compound, In the bottom, above, you can see the STD difference spectrum, and in the bottom, you can see the reference spectrum for Altman compound. As you can see, unfortunately, there is a noise because this is due to the solubility, as I told you, from my compounds, because they weren't soluble at all in T2O. But although we can see some peaks of my ligands here from the aromatic rings, or here, also here for VK1 with lower density, but we can see some peaks of my ligand. Also here for VK2, we can see some peaks. And also here for AD26, here you can see some peaks above of my ligand. Now, the next part was the in silico experiments. The first part of the in silico experiment was the molecular docking. When someone does molecular docking, it has three parts. The first part is that we have the cavity. Here, the cavity is the target, as you can see here from the picture. In our case, it's the 5-lipoxygenase enzyme, or someone can use a protein or an enzyme or different macromolecule. And also we have the possible drug, which is the ligand here. In our case, they are the 4 compounds, VK1, VK2, and etc. And the first part, we have the drug that it tries to insert into the cavity. And then we have the molecular docking. The second part is the selectivity. How selective this drug binds to the active center of the enzyme. If it's very selective, then it means that we have a lot of interactions and binds very strongly to the active center. The third part is the scoring, which is a quantitative representation of the molecular docking, which it shows how much strong these compounds bind to the active center. Now, for the computational part, first we have to prepare the ligand, then we have to prepare separately the protein, and then when we have prepared and the ligand and the protein, induced-fit docking was performed. These calculations, they were performed through Maestro software. Here you can see the results from molecular docking experiments. Here is the enthalpy, is the energy, which means if it's very negative, it means that it binds very strong to the active center. For example, here 8026 binds stronger than the other 3. But these values, they are very low, so all these 4 compounds, they bind very strong to the active center. Also here in the picture, you can see the result of molecular docking, where it's the ATMA compound, the conformation that ATMA compound has after the molecular docking. And these dashed lines represent the interactions between the ATMA compound with the amino acids of the active center, which may be hydrogen bonds or also PP interactions. Here you can see also the interaction between VK1 with the enzyme, VK2 with the enzyme, and also here for AD26 with the enzyme. Here you can see the overall table with the interactions between the ligands with the enzyme, with hydrogen bonds or PP stacking. As you can see, the amino acids, they are quite similar because they bind to the same pocket. Now we proceed to the next part of the in silico experiments, which was the molecular dynamics. After the molecular docking experiments, we took the best result, which means the lowest energy. And the first thing that we had to do, we had to solvent the whole system. Here you can see an example, which the red is the solvent, which is water, and inside you can see the enzyme with the ligand inside. After the solvation of the whole system, the molecular dynamics was performed for 200 nanoseconds. The evaluation of the results was performed through RMSD. RMSD is the root mean square deviation, which it shows the mobility of the ligand inside the enzyme, which generally is a plot. And if in the plot the line is straight, then it means that the ligand it remains stable in the active center of the enzyme. Now I'm going to show you the results to understand better. Here you can see the RMSD for ATMA compound. As you can see here, the red, the line is quite straight, which means that the ligand, it remains stable in the active center of the enzyme the whole of the simulation time over the 200 nanoseconds, and the most of the simulation time it forms an hydrophobic bond with alanine 410. Also here for VK1, the most of the simulation time it remains stable in the active center of the enzyme, and the most of the simulation time it forms an hydrophobic bond with tyrosine 181. Also here for VK2, as you can see, the most of the simulation time also it remains stable in the active center of the enzyme, and the most of the time it forms an iodic bond with isoleucine 673. Finally, 8026, here in the most of the simulation time it remains stable in the active center of the enzyme, and the most of the simulation time it forms an hydrophobic bond again with histidine 372. Now, the last part was to predict the drug likeness of these four compounds. That was performed through SwissADME platform. SwissADME is a very powerful website because someone can draw any molecule that he wants. For example, here we have the benzyl, and someone can see some parameters like the lipophilicity, the solubility, if it obeys some rules, and etc. Here also you can see an hexagon which has 6 parameters. For example, it has the lipophilicity, the saturation, the size of the molecule, if it's very polar, and etc. And if all these lines that you see this in color, in red color, if they are inside the hexagon, then it means that the compound obey all these parameters. It can be used for further experiments. For example, here the benzyl, it doesn't obey the rule for saturation. Also, if someone presses this button next to the couple, which is the target, she can see some possible targets for the compound. For example, here in benzyl, the most probable target is acetylcholinesterase. Now, for my compounds, my compounds, they obey Lipinski's rule of 5, and they are safe and bioactive to be used for further experiments. This is also a very important technique when someone does computational drug design, because some possible biological compounds, they fail to pass the clinical trials due to unfavorable parameters of these parameters. Now, all in all, the main target of my project was to elucidate 4 possible bioactive compounds. That was performed successfully through 1D and 2D NMR spectroscopy. And here also you can see the NMR spectra that I used for the identification. Also, the conformation analysis reveals one possible conformer of its ligand, which it seems that it matches with the 2D NOSI correlation that we observed. Also, from ICD experiments showed some promising interactions between my ligands and the 5-lipoxygenase enzyme. Also, molecular docking experiments show that all these compounds, they bind strongly to the active center. And also from molecular dynamics, it seems that all these compounds, they remain stable in the most of the simulation time inside the active center of 5-lipoxygenase enzyme. Now, these compounds, they have been sent for in vitro now experiments in 5-lipoxygenase enzyme in a collaborative university in Greece, and now maybe future perspective is to perform some extra STDs experiments to other enzymes to compare with this one that we performed. I would like to thank a lot my supervisor and Professor Thomas Mavromoustakos for helping me all these years from my master thesis and now for my PhD thesis until I will finish. Also, I would like to thank a lot the Associate Professor Dimitar Geli for helping me mainly in the computational part of this project. Also, a big thank to Infrastructure, ISIDORE, and others for funding me this trip to the University of Helsinki. Here also you can see some pictures of the university from the laboratory inside of the University of Helsinki. Also, a big thank to the University of Helsinki and the staff inside the laboratory, because they helped me a lot to perform this kind of STD experiments, because I wasn't at all experienced with kind of NMR, unfortunately. And also, I would like to thank all of you for your attention. Thank you.
Thank you very much, Nikitas. So feel free to ask any question that you might have in the Q&A box. In the meantime, I can maybe start with one maybe more general question. I was wondering, so you describe your molecular dynamic and in silico study to identify how the compound binds to your enzyme. But have you thought about confirming this computational approach using experimental data? So you can imagine if the enzyme is not too big to express the protein and label it and do NMR study, but analyzing the protein part and see which residues are involved in the interaction with your compound. So is it something that you have planned or?
Yeah, we have sent now my compounds for in vitro experiments in a collaborative university, but I'm not specialized in this kind of biological experiments, so I don't know, but it's one of our plan. Yeah, good to ask me.
And I was wondering, I could see that you had a lot of pi-pi stacking interaction between your enzyme and your compound.
Yes.
Don't you think that it's a sign of non-specific binding? Would you expect for specific binding more hydrogen bonds or electrostatic interaction or?
No, because they are theoretical calculations, so we don't expect something specific, but it's very normal. It's very normal to see some pp stacking or hydrogen bonds in this kind of computation studies.
Okay. And also, we are now starting to have question. So the first one is a very nice talk, Nikitas. I forgot to mention that. Why did you choose this compound?
Because we collaborate with one other professor in the university. My professor collaborates with the other professor, and they have synthesized these compounds, and they have shown that they have a promising biological activity. And then I took them for further calculations.
Another question here, is one problem possible binding to another enzyme, and would these be detrimental?
Sorry, I didn't understand.
I think it's just asking if one possibility would be that this compound would bind to other enzyme, and if
Yeah, they bind in other enzymes, but now I focused in 5-lipoxygenase because I also performed this STD's NMR spectroscopy in the University of Helsinki. They also bind in other isoforms from lipoxygenase, also in cyclooxygenase, as far as I know, they bind strongly. Acetylcholinesterase also, butyrylcholinesterase, and many enzymes. Yeah.
and so in prospect of using it in vitro studies or in vivo studies, would it be a problem if they bound different enzymes?
This is something that now we have sent for in vitro in this specific enzyme, in 5-lipoxygenase, for in vitro experiments, and we are waiting for the results. Now, I don't know if it's a problem if it binds, because I'm not specialized in the biological part, unfortunately. That's why we send it to another scientist to perform these biological experiments.
All right, and another question now. So do you plan to enumerate analogs for these compounds to improve the activity?
Yeah, why not? Yeah, I can also sketch some possible analogs, and then if they have good results, they maybe will send them to synthesize them and to further then biological experiments.
And I have maybe a last comment for you, Nikitas. When you predicted with your in silico studies the binding energy of your compound, once again, you can have experimental data that would confirm this free energy of binding. For example, you can do ITC or binding interaction measurement. This is also something that you can do to validate your in silico approach.
Okay, thank you very much for saying that. Yeah.
But I can see one last question for you. So binding to other proteins is generally expected for small molecules and result in off-target effect. In the end, there is a trade-off between better therapeutic effect and some toxic burden. So I guess that someone commented on one of the previous questions.
Okay, thank you.
Thank you. Okay, so if there aren't any other questions, well, thank you very much for attending this webinar, and I hope to see you at the next one.